all about mitosis and meiosis.
The main events of the cell cycle include G1, S, and G2 phase, along with interphase and M phase. The M phase, however, takes place during cell division and allows the cell to replicate into 2 new daughter cells.
In sexual reproduction is when two organisms give half of their DNA to the offspring, resulting in an offspring that is half of each parent. Some advantages of this reproduction is that it allows for genetic variation, allowing the species to have a better chance of survival in different environments. However, a disadvantage of sexual reproduction is that it requires two parents, wasting time and energy on finding a mate.
Asexual reproduction is when one organism divides, resulting in a genetically identical daughter cell. Some advantages of this form of reproduction are that it allows the population to produce rapidly and effectively. On the other hand, some disadvantages are that it lacks genetic diversity, so if the offspring gets infected with a disease, the rest of them will as well.
In prophase, the chromosomes condense and attach to centromeres. Imagine a couple of your friends having a big group hug. These are the chromosomes coming together.
In metaphase, the chromosomes line up in the middle of the cell with the spindle fibers on opposite sides in the nuclear envelope. When you see this phase, you think about lining up to go outside. You all stand in a straight line and you have teachers on the side of the line, making sure that you are not misbehaving.
In anaphase, the chromosomes separate, still attached to the spindle fibers, and move to the opposite ends of the cell. When you think of this phase, think of when you and your best friend get a necklace and how they fit together, but when you and your best friend have a piece when you separate.
In telophase, the nucleus reforms around the separated chromosomes, and the spindle fibers are now inside these two nucleuses. Imagine one of your friends moved away, and now you each have your own new group of friends.
Movement of chromosomes. In prophase, the genetic material condenses to create chromosomes, just like cars come together on the same road.
Chromosomes line up across the center of the cell, kind of like how wind lines up in the sky. This explains metaphase.
In anaphase, chromosomes separate and move to opposite ends of the cell, like water rushes from one side of the river to the other.
In telophase, chromosomes spread out into a tangle of chromatin, kind of like how airplanes fly in the sky and the chromatin tangles into a cloud.
In prophase 1, chromosomes condense and crossing over occurs. This is just like mixing ingredients when you're baking.
In metaphase 1, chromosomes line up in pairs, just like how if you were lining cookies up in a cookie sheet. Another example is thinking of the chromosomes that just hugged moving to the center of the cell. These chromosomes are not just sitting in the middle of the cell, they are in pairs, like I said before.
In anaphase 1, the chromosomes are pulled to the opposite ends of the cell by spindle fibers. These spindle fibers attach and pull as if you were grabbing a toy or pulling it towards your body. Another example is if you were splitting dough and moving it to a different place.
In telophase 1, the nuclei are divided into two separate nuclei. This process creates two new cells, along with cytokinesis. This basically means the substance around the cell divides into two, creating two separate cells, or two separate baking batches of cookies.
In prophase 2, the spindle fibers are starting to form, and now you can see them. This phase is different from prophase 1 because the process of crossing over does not happen. The chromosomes in this phase do not hug and share their genetic information. Another example of this is mixing more ingredients into the cookie dough.
In metaphase 2, the chromosomes are in a single file line, unlike in metaphase 1. Just like how a class would line up for lunch, one after the other. The chromosomes come in sets of 1, which means that this cell is a haploid cell. Metaphase 2 has a line leader and a caboose. Just like how if cookies were lining up in a batch.
In anaphase 2, the chromatids are pulled to opposite cell sides. This is like dividing the dough into quarters.
In telophase 2, the nuclei form and the cytoplasm divides, creating 4 new haploid cells. Another example of this is how when you're baking, you will have 4 new batches of cookie dough.
Before mitosis, there is one cell, and after mitosis, it divides into two identical daughter cells.
One way you can think of this is if you had an apple, then you took two halves of the apple to create two new whole apples. Crossing over. This is the exchange of genetic material between homologous chromosomes during prophase 1 of meiosis. One example of this is a puzzle. Switching your pieces creates a new, unique puzzle.
Meiosis begins with one diploid cell and ends with 4 haploid daughter cells.
There are 46 chromosomes in mitosis and 23 chromosomes in meiosis.
This is because mitosis replicates sexually and meiosis replicates asexually. Mitosis reproduces asexually, allowing certain cells to duplicate and heal injuries. This allows for growth and development.
The parent cell is diploid with 46 chromosomes, and the daughter cells are haploid with 23 chromosomes. This is another elaboration on how in mitosis there are 46 chromosomes, and in meiosis there are 23 chromosomes.
Meiosis allows for genetic variation in this species because it reproduces sexually. Thank you.